Welcome to this lecture on evolutionary biology. Today, we are going to explore one of the most profound and magical moments in the history of our planet, biogenesis. This is the breathtaking bridge between a dead chemical world and a vibrant living one. If you have ever wondered how life actually began from non-living matter, you are in the right place. Let us dive in and uncover the origins of life itself. Before we look at the complex biochemistry, let us start with a simple analogy. Imagine you have a giant box of Lego bricks scattered all over the floor. Over time, random shaking causes some of these bricks to snap together into small random shapes. This is much like the chemical evolution that occurred on early Earth. Now, imagine a magical moment where one specific Lego shape suddenly gains the ability to pull other loose bricks from the floor and build an exact copy of itself. This incredible leap from a random collection of parts to a self-copying, organized system is exactly what we call biogeny. Now that we understand the basic idea through our Lego analogy, let us look at the formal scientific definitions. In the study of the origin of life, biogeny represents the critical second major phase. It occurs immediately after chemogeny. Chemogeny was the era where simple inorganic molecules combined to form complex organic monomers and polymers, like amino acids, in the primordial soup. Biogeny is the specific process where these complex organic molecules aggregated and organized themselves into the very first primitive, self-maintaining structures. It is the exact moment chemistry became biology. This transition from chemistry to biology leads us to the creation of the first cellular structures. These early structures are known as protobionts, or protocells. Examples include coacervates or proteinoid microspheres. Eventually, these structures acquired a lipid membrane. This membrane was crucial because it separated their internal chemical environment from the chaotic outside world. Once these membrane-bound structures incorporated self-replicating molecules, like RNA, and began to undergo basic metabolic processes, they officially crossed the threshold into true biological life. They were now alive. Before we move on to the mathematical side of things, let us visualize this incredible transition. On the screen, you are looking at a diagram that maps out the journey we just discussed. On the far left, we have the chemical soup of chemogeny, filled with scattered amino acids and nucleotides. Moving to the center, you can see the protobiont formation, where a protective lipid membrane begins to assemble. Finally, on the right, we reach true biogeny. Here, the first cell is fully formed, complete with self-replicating RNA and basic metabolism. This visual perfectly summarizes the leap into life. Having seen how the first cells formed, we must ask why studying this specific event is so important today. Understanding biogeny is absolutely fundamental because it answers one of the most profound questions in human history. How did life actually begin? By studying this, scientists can understand the absolute minimum requirements for life. This knowledge directly guides our search for extraterrestrial life on planets like Mars or moons like Europa. Furthermore, these principles are actively used today in synthetic biology, where researchers attempt to build artificial cells from scratch to create novel medicines and biofuels. Now that we appreciate the real-world importance of biogeny, let us look at how these early cells behaved. While biogeny is deeply rooted in biochemistry, The defining characteristic that separates these new living protocells from dead chemicals is their ability to replicate and grow in number. Because they can copy themselves, a single successful protocell could quickly colonize the primordial oceans. We can actually mathematically model this very first biological population explosion. By applying mathematics, we can visualize the sheer power of early biological reproduction and see how tiny chemical advantages led to massive success. To model this explosive growth we just discussed, we use the continuous exponential growth formula. The formula is n of t equals n0 times e raised to the power of r times t. Let us break down what each piece means. N of t represents the total number of protocells present at a given time t. N0 stands for the initial number of protocells that successfully formed. The variable r represents the intrinsic rate of replication, which tells us how fast the protocells are copying themselves. Finally, E is Euler's mathematical constant, approximately equal to 2.718. If the replication rate R increases, the population will explode much more rapidly. Now that we have our formula, let us see it in action with a concrete example. Imagine a small tidal pool where 10 successful protocells have just formed. We are given an initial population of 10, a replication rate of 0.5 per day, and a time of 4 days. We substitute these known values into our equation. First, we simplify the exponent by multiplying 0.5 by 4, which gives us exactly 2. Next, we evaluate Euler's number squared, which is approximately 7.389. Finally, we multiply this by our starting population of 10. The result is approximately 74 protocells. This shows how quickly early life could take over an environment.
Having seen a basic population explosion, let us look at a slightly more complex scenario, representing the very first moments of Darwinian evolution. Suppose we have two different types of protocells, type A and type B, competing in the same ocean. Both start with 100 cells. Type A has a replication rate of 0.1 per day. We want to find its population after 10 days. We plug these values into our formula. Multiplying the rate of 0.1 by 10 days gives an exponent of 1. Euler's constant to the power of 1 is just itself. Multiplying by 100 gives a final population of approximately 272 cells for type A. Now that we have the result for type A, we must turn our attention to type B to see how it performed under the exact same conditions. Type B has a slightly better RNA catalyst, giving it a higher replication rate of 0.2 per day. We follow the exact same mathematical steps. We multiply the higher rate of 0.2 by the 10 days, resulting in an exponent of 2. As we learned earlier, Euler's number squared is approximately 7.389. We multiply this larger growth factor by the starting population of 100. This yields a final population of approximately 739 cells for type B. So, we have calculated the populations for both types. This brings us to a beautiful mathematical proof of natural selection. Let us compare the final numbers. In the exact same amount of time, type A grew to 272 cells, while type B grew to 739 cells. Type B has completely dominated the tidal pool. Why? Simply because its internal chemistry was slightly faster. This physically demonstrates that even a tiny increase in replication efficiency during biogeny allows one group of protocells to rapidly outnumber another.
This mathematical result represents the very first instance of natural selection on Earth. To wrap up our lecture, let us review the most important ideas we have covered today. First, biogeny is the monumental turning point where chemical evolution transitioned into biological evolution. We saw how free-floating organic molecules organized into membrane-bound protocells capable of metabolism and self-replication. We also learned how exponential growth allowed these early living populations to rapidly colonize the Earth. Finally, we proved how tiny chemical advantages created the raw material for billions of years of Darwinian evolution. You now have a solid understanding of how life truly began. Thank you for watching.